Hello, wonderful students, and welcome to biology class. In our last class, we discussed about microorganisms, and we said microorganisms are tiny organisms we cannot see with our naked eyes, except with the use of the microscope. We also went further to look at the groups of microorganisms. After then, we look at the carriers of microorganisms. But in this discussion, we shall be looking at microorganism in action. And the subtopic is growth of microorganisms. Before we delve into the topic, let's look at the learning objectives. At the end of the lesson, you should be able to state the different phases in the growth of bacteria. 2. State 4 factors that can affect the growth of microorganisms. And 3. List 3 ways of measuring the growth of bacteria. Bacteria reproduce by binary fission, in which a single cell divides into two. This process is called asexual reproduction. The time interval required for the cell to divide into two is called generational time. This time varies this time interval required for the cell to divide into two is called generational time, and the time varies from one generation to the other. It strongly depends upon nutrient availability, temperature, gaseous requirements, and pH. Let's look at the 4 phases of growth in bacteria. The first phase is the lag phase. The second is the logarithmic or exponential phase. The third is the stationary phase. And the fourth is the decline or the death phase. As you can see from the graph, the first is the lag phase. Then it now proceeds to the one we call the exponential phase. Then the stationary phase, then the decline phase. There are different ways growth can be measured. The first phase is the turbidity method. The second is the serial dilution method. The third is the squared transparent paper or cellophane method. Microorganisms are harmful. At the same time, they are beneficial. We have some beneficial effects of microorganisms. One of it is microbial culture are used to produce enzymes. 2. Yeast is used as a leavening agent in baking industries. 3. Bacteria help to digest cellulose in herbivores. 4. They are used to manufacture amino acids and vinegar. 5. Bacteria are used to process milk into different tastes and flavor. You can see this, you can see all these are the beneficial effects of microorganisms. We also have the harmful effects of microorganisms. The harmful effects of microorganisms are 1. Bacteria cause decay and spoilage of food. If you have a loaf of bread which is exposed to air and bacteria perch on it, you discover that it will cause a decay in the slice of bread. And the second harmful effect is materials like wood, paper, textiles, rubber, and metals are destroyed by microbes. And the third is they cause disease of different types. I will be leaving you with some assignments which you ponder upon and answer them and prepare for the next class. The first is state four different phases in the growth of bacteria. 2. State four factors that can affect the growth of microorganisms. 3. Should be able to explain the three ways of measuring the growth of bacteria. Thank you for your attention. Until we meet again in our next class, keep learning and keep studying. Thank you.